Transformation is the naturally occurring process of gene transfer which involves absorption of the genetic material by a cell through cell membrane, causing the fusion of the foreign DNA with the native DNA, resulting in the genetic expression of the received DNA. Transfection. One of the methods of gene transfer where the genetic material is deliberately introduced into the animal cell in view of studying various functions of proteins and the gene. The mode of gene transfer involves creation of pores on the cell membrane, enabling the cell to receive the foreign genetic material. The main difference is the organisms that are modified by these methods. Transformation is for prokaryote and small organisms, and transfection for eukaryote and particularly animals. Transfection, such as transformation, can be done with some methodologies like the chemical mediation, electroporation, or sonoporation. Also, there are another techniques useful for these processes. For example, the impolifaction, the optical transfection, the lipid-mediated gene delivery, and the use of cationic lipids. There are some important key points to consider to choose an accurate methodology for the process. For example, the size of the DNA fragment that is wanted to insert, the result of the gram stain test made to the bacteria, and even the strain of the bacteria itself. Also, the number of plasmids that is wanted to insert, it will be a big number or a small one, and the efficiency wanted for the process. Other methods. Double digest protocol with standard extraction enzymes. Digesting a DNA substrate with two extraction enzymes simultaneously, double digestion, is a common time-saving procedure. The enzymes start to cut on specific sites while the reaction is still in incubation. High-efficiency transformation protocol using NEP10 beta competent E. coli. It reports to have a great efficiency. The sample containing the plasmid DNA is mixed with the NEP10 beta competent E. coli cells. Then the mix passes through phases similar to the head shock method, but raising the efficiency. Alkaline cations, a method used especially with intact yeast cells, which are treated with the alkali cations to take up plasmid DNA. The most used cations are of lithium, cesium, and sodium.
For the electroporation method, we obtained a total of 7 colony-formed units, where 50 nanograms of DNA were used. We spatulated 100 microliters out of a solution of 1 milliliter. Making a calculation of the efficiency of this reaction with this data, we obtained 1,400 colony-formed units per microgram. Now, for the chemical method, we count 67 colony-formed units for 50 nanograms of DNA. The same amount of volume was spatulated and the total volume of the solution was also the same. The efficiency obtained was of 13,400 colony-formed units per microgram.
Several techniques have been developed to introduce foreign DNA into competent E. coli strains. Electroporation and chemical transformation methods are among the most important ones. Electroporation involves the application of an electric current across the cell, which alters the cell membrane and creates pores in it, allowing the genetic material to go inside it. E. coli are made electroporation competent by thorough washing to remove all medium salts to ensure that the charge is not conducted through the medium, and the electroporation is carried out at low temperatures to minimize heat damage into the cells. Chemical transformation is based on the usage of calcium chloride solution to resuspend bacteria at low temperatures. The cations of the solution interact with the negative charges of the lipidic membrane to create pores and allowing the DNA to go inside the cell through a hydrophilic channel. This is helped with a heat shock, time during which the DNA enters completely to the cell. Electroporation has higher transformation efficiencies and it's more comfortable to manage, but it's more expensive and salt sensitive. Both transformation techniques require the usage of a high purity DNA sample. For the chemical transformation, contaminants like detergents, phenol, alcohols, PEG, and proteins should be avoided. For the electroporation method, the main critical requirement for DNA purity is that it has to be desalted, as electrical shock will be stronger due to the cation interaction of these compounds. The efficiencies obtained during the experiment go from 2 orders of magnitude to 3, while the commercial ones go from a range from 6 to 9 orders of magnitude, meaning that the efficiency obtained during is very low. For the TOPO ligation protocol, an efficiency of 9 orders of magnitude is required for the chemically competent transformation, while for the electrocompetent transformation, higher efficiency is needed as more than 9 orders of magnitude are required. For the low chemical transformation efficiency, the probable causes include impurities in DNA, excess of DNA, and improper cell handling. For electrocompetent transformation, salts and buffers may have inhibited electroporation and caused arcing. Electroporation chambers and DNA were not chilled on ice, or an excessive volume of cells was used.
The main difference between the two processes used in this practice, which were electroporation and chemical means to obtain capable cells, was the usage of salts. In the chemical method, salts are required to neutralize the negative charge of the lipids in the membrane, because they repel the negative charge of the DNA. So, when the membrane is neutralized, the pass of DNA is possible. On the other hand, the electroporation method must be clear of salts. This is because for this method we use an electrical shock to make hydrophobic zones where the DNA can pass. But if we have a little bit of concentration of salts, the electrical shock may kill the cells. The most important parts of the chemical process is the correct usage of salts, diminution in the temperature to stabilize the membrane, and then a thermic shock to modify the permeability of the membrane. And now, for the electroporation method, the most important is to remove all the salts in the solution and the correct usage of the electrical pulse. Well, the efficiency obtained in this practice were of 1,400 CFU per micrograms of DNA for the electroporation method and 13,400 CFU per micrograms of the DNA for the chemical transformation. The efficiency obtained in this chemical transformation was better than the one in the electroporation. This might have happened because not all the salts were removed and some of the cells might have been killed by the electrical pulse.